Good evening. It's the 21st of November 2020. This is Dr. Deepak Natarajan, Director and Head of Interventional Cardiology at Park and Signature Hospitals, Gurgaon, Delhi NCR. Uh, a lot of my friends have asked me to dilate on the Indian vaccine, the completely indigenous one, which is being made by a company in Hyderabad called Bharat Biotech in collaboration with the ICMR. Well, they have done tests on monkeys. Apart from that, they have phase 1 and phase 2 trials, which, however, have not been published as a preprint or in a peer reviewed journal as yet. But the signal is, as per press reports, that the vaccine is safe and it is effective. Therefore, they have begun phase 3 trials. Yesterday, one of the first volunteers was Mr. Anil Vij, who is the health minister and the home minister of Haryana. So kudos to him. The vaccine is an inactivated vaccine. Inactivated means that the virus is procured and it is killed either by heat or by a chemical. The chemical could be a formalin or a propylone. And the killed virus then is attached to an adjuvant. The job of the adjuvant, which could be aluminum hydroxide or quinoline, the adjuvant functions as a booster of immunity. That means it amplifies the immune response in the body. Now, the Indian vaccine is to be given twice. The good thing is that it can be stored in your regular fridge and probably will survive in a regular fridge for 2 to 3 years. But we do not have the required data for me to interpret. However, the Chinese have also made an inactivated vaccine. The company is called CanSino. The vaccine is called CoreVac. They have published their phase 2 trials 3 days ago in the Lancet, and the response to the vaccine was found to be adequate. The side effects were not very many. Just about 30% of the volunteers developed side effects. By and large, these side effects would be headaches, body aches, fever, and fatigability. In this, the Chinese phase 1, phase 2 trials, in the higher 6 micrograms dose, almost 100% developed antibodies. But the quantum of the antibodies was not very high. This was supposedly adequate, but less than that seen in convalescent plasma, which means one is comparing the antibodies produced by the vaccine versus antibodies produced by natural infection. So the inactivated vaccines did produce antibodies to a considerable level, but it was less than the quantum in the average convalescent plasma. The other thing is that the inactivated vaccine produced antibodies in just about 99 to 100% people given the vaccine. Now, as opposed to the inactivated vaccines, where actually the virus, as I've said, is actually it is destroyed, and the body or the shell of the virus is injected into the body, and the purpose of this dead or non-infective dead virus is to trick or dupe the body into making antibodies against the virus, which then prevents one from a future real COVID-19 or a Chinese virus infection. So we wait for the Indian phase 3 trial reports. One presumes they would be out by the end of December or the beginning of January. And once they have been authorized by the regulating agencies, we would be seeing mass vaccination in this country by February or March. One hopes it could be done earlier than that, but making vaccines, testing them, and then actually getting approvals from regulating agencies is not a small job. It is not a small feat. Actually, vaccines take years to develop. For instance, the polio vaccine took 7 years to develop. The measles vaccine took 9 years to develop. The mumps vaccine took 4 years to develop. Chickenpox vaccine took about more than 30 years. And we know that HIV vaccine is still to be produced. An effective vaccine against HIV after almost 4 decades is still not a success, has still not been achieved. However, the COVID vaccines have been developed at record speed within 6 to 8 months. That is really quick. That really is warp speed, and one hopes that they are effective in the real world, because the efficacy may be different from the effectiveness of the vaccine. I will try to explain that. We have data from the Pfizer vaccine that the vaccine was almost 95% effective, which means that if 100 people who were not vaccinated developed the disease, as opposed to that, only 5 people in the vaccinated group developed the disease. To be specific, in the Pfizer study, ultimately 170 people were found to be infected. 43,000 people, 43,000 volunteers were given the Pfizer vaccine. This is actually 2 injections over a 3 weeks period, and what was found is that in the placebo group, there were 162 people who developed the infection as opposed to 8 people who developed infection in the vaccinated group. Also, what is very interesting is that severe infection was found in almost 11 people in the placebo group versus none in the vaccinated group. But the problem with the messenger RNA vaccine is that this vaccine has to be kept at a temperature of minus 70 to minus 80 degrees centigrade, which would be a huge logistic challenge in a country like India, where in summers we all know the temperature soars to more than 40 to 45 degrees Celsius. So it would be a huge task preserving this vaccine. And in fact, even in the US, it would be difficult to distribute this vaccine in the rural areas. As opposed to the Pfizer vaccine, the Moderna vaccine, which has the same technology, well, both use the messenger RNA technology. The Moderna vaccine can be kept in your standard freezer for as long as 6 months, and it can be kept in your regular fridge for as long as 1 month. That means that it can be stored at minus 20 degrees centigrade and between 2 and 8 degrees centigrade. So that is the advantage of the Moderna vaccine, and even this has shown an efficacy of almost 95%. And there have been serious cases, there have been severe cases in the placebo group, but just 1 case in the treated group. So once again, we have an efficacy of 95%. Now, the effectiveness of a vaccine is slightly different, because effectiveness describes the success of the vaccine in reality on the ground. And we know that in reality, people are different. There are people of different sexes, people of different age groups, and also a lot of them will be having other diseases and infirmities. So we still do not know as to how these vaccines will behave in the real world, but one can presume to a large extent that the effectiveness will match the efficacy. If the efficacy is 95%, effectiveness could be, a 90%, 85%, and even as low as 80%. But 80% effectiveness is excellent to control or manage this pandemic. I have already explained to you earlier that the messenger RNA vaccine is a synthetic RNA. Now, this is able to command the ribosomes in your cells to manufacture the spike portion of the virus. Not the entire virus, only the spike is produced. Once the spike is produced, your body then makes antibodies against the spike, and these antibodies then prevent a future or an imminent COVID-19 infection. Remember, the Chinese virus can enter your cell only via these spikes. So therefore, all these vaccines are directed mainly at the spikes of the virus, right? That is very, very important to remember. The messenger RNA is a synthetic RNA bit part which is enclosed in a lipid bubble, and the lipid bubble then ensures that the vaccine can be injected. Once again, the Moderna also has 2 injections almost a month apart. Now, having said that, there is data which has been published by Moderna and which has shown that the vaccine is effective in elderly people also. Even they have a robust antibody response. Apart from that, we have information from the Moderna data that almost they vaccinated about 30,000 volunteers. Of these 30,000 volunteers, about 11,000 were blacks, Latinos, or Hispanics. That means ethnic minorities. Around 7,000 were people above the age of 65 years, and about 5,000 were people below the age of 65 years, but they had either diabetes, extreme obesity, or heart disease. So we have a bunch of people who are not young and who are not very healthy. We have people who are elderly, and we have people who have some chronic disease. So therefore, the Moderna vaccine data is quite impressive. Pfizer has already applied to the FDA for authorization yesterday, and hopefully the permission will be given by the mid of December. So we have discussed the inactivated vaccine or the killed vaccines, and examples of inactivated vaccines otherwise are the hepatitis A vaccine, influenza vaccines, polio vaccines, and the rabies vaccines. So apart from that, we have discussed the messenger RNA vaccines made by Pfizer and by Moderna, and then we also have what is called the DNA vector vaccines. Now here again, this is a gene vaccine, and the genetic material or the genetic code which will coax or prompt the ribosomes in your cells to produce these spikes is carried by a common cold virus called the adenovirus. Now this virus is actually altered in the laboratory to make it not infective. The virus is essentially has become ineffective, and then this virus acts like a taxi or like a cargo plane which ferries the genetic material, the DNA material, which penetrates the cell and then instructs the ribosomes to make the spikes of the virus. And once again, the entire virus is not created. It is just the spikes which are recognized by the body, and the body presumes that this is the enemy and this is the actual virus. But the antibodies produced are adequate, have been found to be adequate, and these antibodies again will help the person concerned to fight back or prevent a future China virus infection. So we have now spoken about the DNA vector vaccine, we have spoken about the messenger RNA vaccine, and we have spoken about the inactivated vaccine. Now the Oxford vaccine, which is being made in collaboration with Astra Zeneca, will be tested in India also. The phase 3 trials have begun. They already have done monkey trials, and the animal trial has shown that the vaccine was capable of preventing virus entry into the lungs of the monkeys, but did not prevent the presence of the virus in the throats and the mouths of the monkeys. Now, the clinical significance of this is that the Oxford Zeneca vaccine may convert a serious illness into a mild illness. It probably is incapable of preventing infection, but it can stop serious infection. The other thing is that the antibodies produced, though robust, are not very high. The quantum is not very high. Once again, the quantum of antibodies produced is somewhere in between the antibodies produced by the inactivated vaccines and by the messenger RNA vaccine. And the AstraZeneca group has published a paper in which they have checked their vaccines in elderly people also. This was published again 3 or 4 days back in the Lancet. They had 3 groups of volunteers. First was the young group between 18 and 55 years. They had a group between 55 and 70 years. And then they had the third group above the age of 70 years. What was seen was all 3 groups produced robust antibodies against the vaccine. But interestingly, the adverse effects were seen lesser in the, or rather least in the elderly group, that is above the age of 70 years. Adverse effects were seen in about 85% of the younger group, that means below 55 years, in 75% of the group between 55 and 70 years, and in 63 or 65% of people in the group above the age of 70 years. However, one has to realize that the people above the 70 years were usually in their early 70s and were rather fit. Most of them did not have any serious disease. So having said that, I must now mention the other company, that is Johnson & Johnson, which also has a similar kind of a vaccine, which is that the virus, the gene which is going to order the cell to make the spike is carried by the common cold adenovirus vector. And the important thing in the Johnson & Johnson vaccine is that this has 2 protocols. One is a single injection, the other one is a double injection. That is just like the Moderna, Pfizer and the Oxford vaccine and the inactivated vaccines. This has also 2 injections, 3 or 4 weeks apart, plus a single injection. So the one advantage is the single injection. The second advantage is that the antibodies production is very high. It is as good or higher than the messenger RNA vaccines and definitely higher than the AstraZeneca vaccine. Plus, the important thing is that the Johnson & Johnson vaccine can be stored for almost a year in your standard freezer and for almost 3 months in your fridge. So that would be a big advantage. The AstraZeneca vaccine does not require a freezer. It can be stored in a fridge. So that is one logistic advantage. And I think I've already told you that the AstraZeneca vaccine is being administered to volunteers in India. And around 30,000 or 35,000 volunteers are taking part in the phase 3 trial. And the results should be out within a fortnight or 3 weeks. Because there is a leak. Now, I don't know whether the leak is deliberate or whether it is a genuine leak, that the NHS and the British government is very keen to begin vaccinating next month. They will start vaccination first in health workers, in people who live in care homes, the elderly, and the staff which looks after these people who live in care homes. They would get the vaccination as early as mid-December. And towards the end of December, people above the age of 80 years, followed by people above the age of 70 years, will be vaccinated. And then in January and February, people below the age of 70 years, especially people who carry diseases like diabetes or heart diseases, would be vaccinated. And the ultimate plan is that by spring, that is by March, they would have the entire population of the UK vaccinated from the ages of 18 years above. And of course, if they use the Moderna or the Pfizer or their own Oxford vaccines, then they would need 2 injections. And when we bear in mind that the British population is about 65 million people, so they will need 130 vaccines, but they have already ordered for more vaccines, more vials than this number. They have already ordered. They have placed orders with the Moderna, they have placed orders with the Pfizer, and of course they have placed orders with AstraZeneca. And AstraZeneca is making a lot of these vaccines in India, in Pune, in the Serum Institute, which is one of the foremost factories for making all kinds of vaccines in the world. Now, I come to the fourth kind of vaccine, and this vaccine here, this is not inactivated. This is also a pretty new technique. This is called a nanoparticle viral protein vaccine. Now, in simple English, this means that they do not use genetic material to induce the human cells to make spikes. They actually create these spikes, right? They have ready-made spikes, and these ready-made spikes are injected. And once the ready-made spikes are injected, there is a very strong antibodies response. They already have published, they already have a preprint of their phase 1 trial of about 135 volunteers, wherein 2 injections with this nanoparticle vaccine, which is being made by Novavax, had elicited a very strong response. In fact, the antibodies produced were 4 times that seen by good convalescent plasma. This means that the antibodies produced by the artificial virus was 4 times more than that produced by natural infection. Now, the spikes are produced in the cells of moths, and once the spikes are produced, they are put on an artificial platform, and this is then combined with an adjuvant, and this adjuvant is not alum, nor is it quinolines. This is actually from the plant kingdom. It's called matrix M1, and this is supposed to be also a very strong immunity booster. So the Johnson & Johnson company also has phase 3 trials going on in a variety of countries. They in fact have almost, I think, 90,000 people, volunteers for their phase 3 trial, and one can presume that their vaccine too would be effective and would be safe. And the biggest advantage would be that the logistics would be much easier than the messenger RNA vaccines. But I don't know whether the Indian government has placed an order with Novavax, but the important and the good news is that Novavax vaccines will be made in India, massive amounts. The company has announced that maybe almost 2 billion vaccines would be produced in India next year. So 2021 would be called the vaccine year, the COVID vaccine year. And because all these vaccines will come into play, and at this point, I must tell you that the vaccines come with certain caveats. These are almost like cliches now. We do not know whether these vaccines prevent asymptomatic infections. We know that these vaccines by the Moderna and the Pfizer trials, they do prevent symptomatic infections. But if they do not prevent asymptomatic infections, then that means they do not prevent transmission. And if you get infected after being vaccinated by the virus and you do not produce any symptoms, then that could be a paradoxical problem, because you would then lower your guards. Maybe you would not wear your mask, maybe you would not maintain your distance. So therefore, this is a problem which has to be sorted out. We must know whether these vaccines are capable of preventing transmission. The other problem is, if the vaccines do not prevent transmission, then they would have their role in producing herd immunity would be severely diluted, because you have a huge bunch of people who have been talking about vaccines and the fact that vaccinations will induce or produce herd immunity. I have my doubts. If the vaccine is incapable of preventing infection, then the vaccines would not be able to produce herd immunity. But then this is tempered with the knowledge that in most cities of India, where these tests for antibodies have been done, the seroprevalence rate is pretty high. In Delhi, 25% of people have antibodies. In Srinagar, almost 50% of people have antibodies. In Pune, again, 50% of people have antibodies. There is now information that of the 50% people in certain areas of Pune who have antibodies, 85% of these people have neutralizing antibodies, which means that they have antibodies which would definitely prevent a future infection. So that is good news. So what I'm trying to imply to you is that even if the vaccines are not the magic bullets to achieve herd immunity, we already are inching towards herd immunity because a very large number of our populations have already been infected and we already have antibodies. And apart from that, the fact that we have the lowest mortality rate, one of the lowest mortality rates in the world as per million. Even today we have a mortality rate of about 92 or 93 per million. The Americans have it at 750 per million. England also has it at 750 per million. Spain has it at 850 deaths per million population. So our mortality is very low, which means that there is a very, very strong probability that we already have antibodies induced by earlier respiratory infections and which now help us in preventing COVID-19. And this has been reported, has been documented in a number of papers from the very, very good academic centers that people with earlier common cold virus infections do develop antibodies, especially T cells, which can cross-react against the Chinese virus. So this is a very important piece of information which you should keep in mind. Now we move on and of course there's another point is that the other caveat is nobody knows what will be the duration of the immunity provided by these vaccines. We do know that definitely antibodies last for around 7 to 8 months after a COVID-19 infection, regardless of the fact whether it is a mild or whether it is a severe infection. Antibodies are there, and this data has been presented by a very good immunology center called the La Jolla Institute of Immunology in the United States of America. This is still in the form of a preprint, but the data is very good, and it is very strongly suggesting that that antibodies should last at least for 7 to 8 months after an infection with COVID-19. And it also mentions the fact that T cells produced by previous other virus infections last many, many years. And it gives the example of SARS. SARS is the severe acute respiratory syndrome, and by coronavirus number 1, which came up in 2003, those people still have T cells against that virus as long as 17 years after the first infection. Now, if you have T cells, let's say for 2 years after the Chinese virus infection, that does not mean that it will necessarily prevent the infection, but there is a very, very strong chance that even if you do get infected, these antibodies will dilute or reduce the clinical impact of the Chinese virus infection. So now we will move on to another subject, that is the PCR test. Now I will talk about the PCR test, and this is very important for you to know because it is mandatory that when you get an Rt-Pcr test done, that to rule out a false positive test, you must know the cycle threshold at which the virus has been picked up. Now, what is a PCR test? A PCR test basically doubles that genetic material, that microscopic material. It doubles it and then doubles it again and then doubles it again and then again and again and again and again and again and again. So you carry on doubling till you get millions or billions of pieces, which can then be detected by your system, right? So these doublings are called cycles. So therefore you can have a cycle threshold of 10 or 15 or 20 or 25 or 30. So the important thing to realize is that if you can pick up the virus at a low cycle threshold, that means you have a big viral load. That means you have a lot of virus in your body. And if you pick it up at a higher cycle threshold, that means after doubling so many times, then either you do not have the virus, or if you have the virus, that virus is not viable. That virus is already dead. That is not virus, that is just genetic material. In fact, you are picking up only fragments or bits and pieces of the genetic material of the Chinese virus. Now, I am talking about the PCR thing because very recently, on the 11th of November, that is just about 10 days back, an appeals court in Portugal made a ruling which is very, very, very important, and which has not been covered by the mainstream media anywhere in the world. But I will provide you the link. And the ruling is that in a person who is healthy, or rather a person who is asymptomatic, a PCR test by itself is not infallible. And because the PCR test is not infallible, you should not isolate or quarantine a human being. Your liberties should not be constrained because of a PCR test. Now, I will give you the background of this ruling. It so happened that 4 Germans flew down to Portugal. Before they came to Portugal, they tested themselves by the PCR test in Germany. All 4 were negative. We don't have their names and we don't have their sexes. But these 4, when they landed in Portugal, were tested again, and it was found that one of them was positive by the Portuguese PCR test. And because this one person was positive, that person was isolated, and the 3 people accompanying this person were quarantined. So therefore, they made an appeal, and the health ministry of that territory, of that island in Portugal, also then appealed to the court. But the court, in its infinite wisdom, their lordships, it was a 2-bench ruling. They made, I think, an incredible ruling, because they cited data, and citing data, they made it clear that it should be mandatory that you mention the cycle thresholds of the PCR test. And I think the reason they said this was that in Portugal, the PCR tests do not carry the cycle thresholds at which they become positive. So what their lordships said, and they cited a very, very good study which I have already spoken about. This is a French study which was published in an infectious disease journal in the US. This is from France. The lead author is a person called Rita Jafar. And what this study says is that everyone who carries a PCR test positive, or is presumed to be carrying the virus, as per the PCR test, would not necessarily be carrying a live or a viable virus. It is not necessary that that person is infective to the people around him or around her. And what they did was, their study then describes 180,000 people who were tested by the Rt-Pcr test. Of these, 13,400 came out to be positive. Of these 13,400 people, they had about 3,400 people in samples. These samples were cultured in the laboratory. Now this means that the specimen was put in a petri dish in the laboratory, and they waited to see whether the virus there began to multiply. If the virus multiplies or replicates in the laboratory, that means it is culture positive. And if it is multiplying, that means the virus is alive, right? If that specimen, when put in the laboratory, does not multiply or does not replicate, then this is not a viable virus, and this person is not infective. Now, what they did was they correlated the culture positivity with the cycle threshold of the PCR tests. Now, you've got to be, listen to me. You've got to listen to me very, very carefully. What they found out was that when the virus was detected at a cycle threshold of 25, which means the material had been doubled 25 times, right? Then in 70% of the specimens, the culture was positive. 70% of these specimens could multiply or could replicate in the laboratory. 30% of the specimens did not replicate, which means 30%, one third of the cases were not infective. When the cycle threshold went up to 30, now 80% of the specimens were not able to replicate. Only 20% of the specimens multiplied in the laboratory or could be cultured. So this means that if you have a PCR test with a cycle threshold of 30, then the chances of the virus being not viable, the chances of the stuff which you are testing being just a fragment of dead genetic material is as high as 80%. Only in 20% would the virus be viable. Now you take the cycles to 35. At 35, only 3% of the specimens were viable. Only 3% of the specimens could be cultured. 97% of the time, the virus was dead. It was non-infective. I would repeat this. At a cycle threshold of 25, 70% of the specimens were positive, were alive. At a cycle of 30, just 20% were now positive or actually viable or could be cultured. At a cycle of 35, just 3% were alive, and 97% of the specimens were non-viable. They were just fragments of dead genetic material. And anything above 35 cycles was a big no-no. There was no virus at all. There was no genetic material at all. So now, citing this study, the judges in the Portuguese appeals court said that, if you get a PCR positive test positive at a cycle threshold of 25 or below, then that would suggest an active infection. Anything above 25 could be anything, and above 30 would be highly improbable. Now this is a huge, a massive ruling, and I wonder when this news will spread around in the world, or whether it will spread around at all. But my humble submission is that if you check on a PCR test, or if you get a PCR test done, ensure that you know that you have the information of the cycle at which the virus appeared. So remember, if the cycle is below 30, then there is a strong chance that you are having the virus, and if the cycle is above 30, then the chances of there being of an infective virus are quite remote. You will be scandalized to know that in the US again, the cycle thresholds were not displayed all these months, and in fact the cycle thresholds were kept at almost 35 or 40. So there must have been a whole lot of false positive tests with the Rt-Pcr tests. And in fact, I think in the month of May, I had said that the PCR test is certainly not the gold standard for the diagnosis of COVID-19. So what is the gold standard? The gold standard should be that if a person you presume has been infected, you take the specimen from the nose or from the throat or wherever, or from the saliva, and you ensure that apart from the PCR test, or in addition to the PCR test, that specimen can be cultured in the laboratory, or you look for what is called a subgenomic RNA, which indicates that the virus which you have picked up is multiplying. But these, of course, are difficult tests to do, and I doubt if anyone is doing them outside academic laboratories, and it would be difficult. So therefore one has to keep a simple fact in mind that there will always be many false positives, especially in the community, and there will also be many false negatives. But in a hospitalized patient, in a patient in a hospital with a pneumonia, which is seen on an X-ray or a CT scan, then the specificity or the predictive value of a PCR test is high. But testing in the community, like you go around testing in Delhi, they're saying that they're doing 60,000 tests per day, or at least they've done 50,000 tests a day last Tuesday, and almost 25% of these are PCR tests. So you should be informed that a large, a significant number of these tests could be falsely positive. The PCR tests must be accompanied by the cycle thresholds. Now, I am mentioning this because there is another very good study which has been published just 2 days back, once again in the Lancet, and this is a systematic review and a meta-analysis of 79 studies of SARS-CoV-2, and what they found was very interesting. They found that the patient concerned could be shedding the virus or carrying the virus in the mouth and in the throat for almost 17 days. They could be carrying it in their lungs for almost 15 days, and the stool also has the virus for almost 17 days. And the upper limit is that if you have the virus in your nose or in your mouth, then the upper limit is that you could carry it for almost 3 months. It can be carried for 2 months in your lungs, and it can be carried for almost 4 months in the stool. Now that seems to be very scary news, that a person who's got infected will carry the virus for 3 months in his nose and in his mouth. No, the point is, and which these people have also said, the researchers of this article published in the Lancet, what they have said is that without fail, regardless of the amount of virus in the human body, or wherever it is, whether it is in the upper respiratory system, or whether it is in the lower respiratory area, or whether it is in the fecal matter of the person concerned, the important thing is that after 9 days, the virus ceases to be infective. The virus is now more or less dead, right? So this is something very important, that in case a person is found to be by an Rt-Pcr test to be positive, and we presume or we know that the PCR test is definitely correct or accurate, then even that person, regardless of the status of his symptoms, regardless of how much virus he is carrying, after 9 days, after 8 to 9 days, the person is not carrying live virus. The person has ceased to be infected. So therefore, scientifically or logically, there is no need to do an Rt-Pcr test again. And this protocol of doing 2 Rt-Pcr tests after 14 days or after 20 days is quite unscientific, as we know. Of course, we know much more now than we knew 6 or 7 months back. But the fact still remains that an Rt-Pcr test is not infallible. It is not the gold standard. It can make mistakes. Mistakes can be made at many levels, at the point of collection, at the point of storage of the specimen, during transportation from the storage point or from the collection point to the laboratory. Even in the laboratory, it could be contaminated from people or air or substances carrying the virus, and there could be chances of false positives. Of course, there are also false negatives that also has to be kept in mind, so one must use common sense when you are tackling these people. And I talk about the virus and the infectivity or the replication period because all data points to the fact that multiplication is maximum in the first few days, and especially on the day of onset is the peak of multiplication. That means that is the time when the person concerned is most infective. to people around her or him. And I say this because the treatment or the management of COVID-19 is now less problematical because now we have more information, and it can be divided, as far as I'm concerned, into two phases. The earlier phase, which is when the virus is multiplying, and then the second phase when the virus has ceased to multiply, but there are some unfortunate people who kick or trigger or spark a very an exaggerated immune response called the cytokine storm, wherein the antibodies produced or the immunity produced in the body acts not only against the virus but actually against the body itself. So that is the later phase. So therefore, all medicines which are considered to be antivirals, that means medicines which prevent the multiplication of the virus or attack the virus directly, should be given as early as possible. This can be a problem, but there are some medicines, and I must, I will enumerate them. One is remdesivir. Definitely, despite the fact that the WHO says that it should not be used in a hospital, but the WHO data is very heterogeneous and full of loopholes. I would be inclined to accept the data from Gilead, the makers of remdesivir, and they had published a double-blind, placebo-controlled, randomized trial which showed that there was a significant reduction in recovery from 15 to almost 10 days in people who were confirmed cases of COVID-19. So therefore, right in the beginning, if you have remdesivir, you must get the injections. Just 5 injections would do. The second thing is, of course, monoclonal antibodies. These have been, I think, given emergency permission by the FDA. They were used in Donald Trump also. These should be used immediately in the first 2 or 3 days. Of course, these are not available in India. They are very expensive and difficult to produce. But the fact is, 2 companies are making them, Regeneron and Eli Lilly, and both have their own kinds of monoclonal antibodies which are antiviral, which attack the virus directly, and by doing that, they improve clinical outcomes. The third medicine would be an antidepressant, fluvoxamine. Fluvoxamine is an SSRI, that is a selective serotonin reuptake inhibitor, and this medicine is freely available in India. Just about any chemist would have it. The medicine is a tablet. You need to take 3 tablets a day for about 10 or 14 days, and the dose is 100 milligrams 3 times a day. I mention this tablet because a double-blind randomized trial has been published in JAMA, which showed significantly improved clinical outcomes with this antidepressant, and this works through the SIR mechanism. The SIR mechanism is responsible for triggering the cytokine storm. So by stopping the SIR mechanism, you prevent a cytokine storm, and therefore you prevent the deterioration of patients of COVID-19. But again, this medicine should be given much earlier, and it can be given early because it is a tablet and is available freely in India, and the drug is quite cheap. It is in fact very cheap. The fourth medicine would be interferon. Interferon again has been shown in a double-blind randomized placebo-controlled trial to improve clinical outcomes significantly, and this has been published in the Lancet, and this is echoing another study published some months ago wherein the interferon was given as an injection, and that was also a randomized trial which showed that interferon reduced recovery time from some 12 to 7 days. Coming back to the study which was published a few days back, with interferon this time has been given by nebulizer, and the action of interferon is that actually it is the first defense mechanism in the body. Once it is produced, it is antiviral, it attacks the virus directly, and therefore improves clinical outcomes in patients with COVID-19. But one has to wait for a larger randomized trial before one fully endorses nebulized interferon, or oral fluvoxamine. And apart from these drugs, of course, in the very few unfortunate people who have to land up in hospitals and who are not improving and who require supplemental oxygen or sadly need a ventilator, we have a large randomized trial which has shown that there is a significant 30% reduction in mortality in patients on ventilators. Also, there was a significant 20% reduction in mortality in people who needed supplemental oxygen, but with a drug called dexamethasone, which is a common steroid, once again freely and cheaply available in all cities and all towns of India. The important thing to remember is that steroids should not be given in mild cases because they dampen the immunity system, they may prove to be harmful. As I said, the first half of the infected period demands antivirals. In the few people who go on to develop the exaggerated hyperimmune response, the best drug would be dexamethasone, and that has been confirmed in a large randomized trial. And the other drugs would be interleukin inhibitors, and that also has been shown in a large observational study to be effective. And there's a third drug which has been published, the data has been published very recently. This is baricitinib. Baricitinib is a JAK inhibitor. JAK is a Janus kinase system. The Janus kinase system again is a source of letting off the cytokine storm. So by giving a JAK inhibitor, one can abort this cytokine storm and thereby help in patients' recovery. And this study, this observational study, and which has been published, has shown that there was a 71% reduction in mortality in patients who were on ventilators. Also, these people had an average age of 81 years. It was a small study, but the signal is very strong. So what I'm underlining is, I hope you get the drift. What I'm trying to tell you is that we have a lot of ammunition against this pandemic. So there is no reason to be despondent. Please do not be morose. Please do not be unhappy, because we will be turning the tide. We have basic measures like hand hygiene, personal hygiene, wearing a mask where it is absolutely essential, maintaining distances and avoiding crowds. And if we go to the market, making sure that we are at least 3 to 4 to 6 feet away from the other chap. Right? Those things still matter. And even if you get the vaccine, they will continue to matter. You will have to continue to employ these techniques. Apart from this, we also have medicines which have been proven in randomized studies to help patients recover, and also mortality has been reduced. And these are remdesivir, you have monoclonal antibodies, you have fluvoxamine, you have interferon, and in the few people who really get sick, you have dexamethasone, which is freely available, and you have the JAK inhibitor, that is baricitinib, and you have the interleukin 6 tocilizumab also, which is available. Again, it is available in India. Of course, it is very expensive. But what I am trying to tell you is that we have now a variety of medicines by which we can tackle this pandemic. And I'm sure there are scientists, there are researchers who are working with other medicines and other forms of therapy to combat this pandemic. Now that Diwali is over, I hope better sense prevails, and that the public in Delhi comes back to maintaining some distance, wearing a mask in crowds, and of course washing their hands as frequently as possible. Because remember, even though we make it a vaccine, there is a very strong chance that the vaccine may not prevent an asymptomatic infection, or it may at best convert a serious illness into a mild flu, which means that you may have an asymptomatic infection, you may be a source of infection for your family, for your relatives, for your loved ones, and for people around you, for your colleagues, because you may be having the asymptomatic infection, and therefore there is a chance that you may be still required to wear masks, you may be still required to maintain distances. So vaccines will not be a panacea. They will be an important component against the pandemic. I hope you have got some information. I have tried to simplify matters. It is difficult to simplify complex medical papers, because I have to speak on immunology, virology, on microbiology, clinical medicine, so it becomes a bit difficult. And then I have to simplify all this stuff so that you can understand what I am saying. I have noticed that there are other people making videos, there are people across the world, but somehow they are unable to crystallize or simplify the basic fundamentals regarding this pandemic. So if you have enjoyed this video, or even if you have liked it partially, I thank you. Please do press the subscription button, press the like icon, and press the bell icon, and do share this video with as many people as you can. Thank you very much, and a very good night.